Hi everyone, welcome back to Untitled Biology Discuss. Today we will discuss about the Morgan's experiment to prove the linkage. As you know that linkage, what is the meaning of linkage? Linkage means if we see some chromosome, the chromosome bears some kinds of genes. Several numbers of genes are present over a single chromosome, and the genes which are present over the same chromosome, that is called as linked genes. And inheritance of these linked genes, or the ability of these linked genes to transfer to the next generation, that is called as what? That is the linkage. Okay, as you know that this linkage was first of all talked by Bartesson and Punnett, but this was also proved by the Thomas Hunt Morgan. Okay, let's see what happened the Morgan's experiment. And if you see Morgan's experiment, then Morgan chose Drosophila melanogaster as his experimental material. Okay, so Drosophila melanogaster, or the fruit fly, took as his experimental material. Okay, so fruit fly, if you see fruit fly, then in the fruit fly there is presence of 2n is equal to 8, means there is presence of 8 number of chromosome in the diploid organism. And out of this 8 chromosome, 6 are the autosomes, 6 are the autosomes and 2 are the sex chromosomes. Okay, these 6 autosomes, these are also called as your somatic chromosome, whereas this sex chromosome, that may also called as heterosomes, or it may also called as idiosome. Okay, or simply this is called as sex chromosome, or sometimes it may also call as allosome. Okay, so if we see the sex chromosome in case of Drosophila melanogaster, as you know that Drosophila melanogaster, there is one male individual and female individual. If we see the male individual, there is presence of XY type of sex chromosome, means one X chromosome and another Y sex chromosome is present in case of your male individual. Whereas in case of your female, there is presence of XX type of sex chromosome. Okay, so if we see here there is presence of 2 different types of sex chromosome. One is X, another is Y. Here there is presence of 2 same type of sex chromosome. Both are the X chromosome. Okay, so this condition, that is called as, or this is called as what? This is called as heteromorphic condition. This is called as heteromorphic condition. And this is called as homo means same. This is called as homomorphic condition. Okay, this is heteromorphic and this is homomorphic. So if we see some genes that are associated with this X chromosome or Y chromosome, then what happens? Let us see. If you see XX and XY type of sex chromosome, okay, suppose the X chromosome, suppose these are the 2 X chromosome, okay, and this is X, and here suppose these are 2, this is X chromosome and this is Y chromosome. Okay, here in case of your female, the homologous chromosome of this X chromosome is this, and the homologous chromosome of this X chromosome is this, means there is presence of one pair of homologous chromosome. But if you see male individual, in case of male individual, there is presence of X chromosome and another Y chromosome. That means the homologous partner of this X chromosome is absent in case of your male Drosophila. Thus, what happened? Suppose a gene A. Suppose you consider a gene A, then the homologous partner of this gene A, that will not present on the Y chromosome. Suppose capital A. As you know that always gene expression pair condition. A gene is having 2 alleles. Okay, so one allele there is A, another allele there is capital A. Okay, suppose this is a homozygous condition. But if you see here, in case of your XY type of sex chromosome, here in case of male Drosophila, the X chromosome that is having capital A, but here the capital A or small a both are absent in case of Y chromosome. The homologous partner of this allele A that is absent in the Y chromosome. And suppose we are considering about the recessive B gene. Suppose we are considering about the recessive gene B. Okay. Thus the homologous partner of this B gene will not present over the Y chromosome. Why this is not present? Because the homologous chromosome of this X chromosome is not, the Y chromosome is not the homologous chromosome of this X chromosome. The Y chromosome that is not the homologous partner of this X chromosome, or X chromosome is not the homologous partner of your Y chromosome. Thus, what will you often see here? Here, a allele, as you know that according to Mendel, always its factor is expressed in the pair condition. Okay, but if you see here, gene A will express in unfair condition. Gene A or gene B, here both will express in the unfair condition. Okay, and this condition that is called as what? This is called as hemizygous condition. This is called as hemizygous condition. What is hemizygous condition? As you know that all the alleles are expressing in the pair condition. But if you see here, in case of your male individual, in case of your Drosophila, there is absence of homologous partner of these genes. Thus, only one allele will express here. That's why this condition is called as hemizygous condition. Okay? If we see the recessive gene, here this is the recessive gene, recessive B gene. Recessive B gene, that is the recessive gene. As you know that recessive gene always expressed in the pair condition. Recessive gene always occurring in pair condition, and always this recessive gene also expressed in only homozygous condition. Recessive B gene that will only express in this condition. Okay, but if you see here, this recessive gene has the ability, okay, this recessive gene has the ability to express in unfair condition or heterozygous condition. And you know that this is always expressed in homozygous condition. Okay, always this is expressing in homozygous condition. But if you see here, it will express in the heterozygous condition. Only one will be there, but that B gene also expressed here. Okay, that is called as what? That's why this B gene, this recessive B gene will be called as pseudo-dominant gene. Pseudo-dominant gene. This will be called as pseudo or false. This is a false dominant gene. Okay, let's see if we go for the experiment. Let's go to the experiment that is done by Morgan. Okay, Morgan choose 3 different characters. He chose 3 characters. What are these characters? One is the body color. Okay, another is the eye color. Another that is wing size. Okay, so these are the 3 different characters that is taken by Morgan. Morgan found 2 body colors in case of Drosophila melanogaster. One is the brown and another is the yellow. Where the brown is the dominant variety and this is the recessive variety. This is the dominant variety, whereas this yellow variety that is the recessive variety. And here in case of your Drosophila melanogaster, as you know that the recessive gene that is called as pseudo-dominant gene. The recessive gene that is called as pseudo-dominant gene. Thus, Morgan took this brown variety as Y plus and this recessive variety as Y. Okay? And if you see the eye color, then there is 2 eye color. One is the red, another is the white. And what is the recessive gene? And red is the dominant. Thus, the small w flaws that is representing red and small w that is representing white. And wing size, there is 2 wing size. One is the normal wing and another is the miniature or very small. Okay, this is the recessive, this is the dominant. So that is took this normal wing as M plus and this is as small m. So these are the symbol that is used by Morgan and the characters which are selected for his experiment. Morgan found that all these three characters or the genes which are responsible for these three characters that are associated with the X chromosome. Okay, so the genes which are responsible for these characters are associated with the X sex chromosome. Okay, and during his experiment, Mendel performed three different dihybrid cross. Okay, one dihybrid cross between this body color and eye color. This is the dihybrid cross 1. Then with eye color and wing size, this is the dihybrid cross 2. Then he selected this 2, that is body color and wing size, and this is the dihybrid cross 3. Okay. So first dihybrid cross, that is performed between the body color and eye color, whereas the second, that is performed with the eye color and wing size, and the third dihybrid cross, that is performed by body color and the wing size. Okay, so let's go to the first dihybrid cross, that is between the body color and eye color. As you know that in the dihybrid cross we have to choose the two characters. Here the character 1, that is the body color, and the eye color. Okay, let's go for the crossing between these two characters. Let's see dihybrid cross 1. In dihybrid cross 1, Morgan selected two characters. One is body color and another is eye color. For body color there is two varieties. One is the brown, another is the yellow. For eye color there is two varieties. One is red, another is white. Brown is the dominant and yellow is the recessive, and in the here we took brown is the Y plus and yellow is the Y, and red is the W plus and white is the W. Okay, so Morgan select one homozygous recessive female parent, one homozygous recessive female parent. Homozygous recessive means this is the recessive one, this is the recessive one, that is yellow body color and white eye color. He crossed this with the heterozygous dominant male individual, okay, or male parent. That is heterozygous means here the male, that is the heterozygous, because X and Y chromosome are there, and we know that the body color and eye color, that is associated with the X chromosome. Thus dominant, this is the dominant, this is the dominant, means brown and this is the red eye color, brown body color and red eye color. Yellow white, this is X chromosome, because this is the female parent, this is the female parent, this is the male parent. Thus here there will be presence of XX chromosome, and yellow for the small y and white for the small w, brown for the small y plus red for the small w plus. Thus the chromosome will having YY genes or the YY alleles, and here this is WW, small w, small w. It will cross with the male individual, means X and Y. X chromosome is having Y plus and W plus. But the gamete will form according to principle of segregation, both will separate. Both will separate. So this will X, this will X, this is Y, this is W, this is small y, this is small w. And here also there will be separation of these two chromosomes, that is X and Y. And here this is X is having Y plus and this is having W plus. If this will cross with this, what happens? If this will cross with this, then what will be the result? Let's see. X. If this and this cross, then small y, small w, small y plus, small w plus. Okay. If it will cross with these, then what will happen? X and y will form, and this is small y, small w, and here that is absent. If it is crossed with these, okay, then what will be the result? X, and here this is the small y plus, small w plus, and this is small y, small w. And if it is cross with these, then what will happen? This is the result. This is x, this is y, and this is small y, small w. So this is the female parent, this is the female parent, this is the male parent, this is the male parent. Okay. So if we see the genotype, what will be the phenotype? Then this is out of these two, which one will express? Small y, small y plus will express, that is for the brown, and this is for the your red. So this is brown, red. Out of these two, one is yellow, small y, that is for the yellow, small w, that is for the white. Here, small y plus, small w plus. Small y plus, that is your brown, and small w plus, that is your red. Okay, because out of these two, the small w plus will express, and this is the dominant variety. And out of these two, small y plus, this is the dominant. And here, this will become same, that is yellow and white. Okay. If you cross between one of the male with the one of the female, then we will get the F2 generation ratio. This is male individual, this is female individual. We have to cross between these two individuals. Then let's see what will be the result. So we have to cross between the one female individual and male individual. So this is the female individual XX, and X is having small y, small w, and small y plus, small w plus. Okay, this is brown, red. This will cross with the male individual, that is the X and Y chromosome, and one X chromosome is having small y, and another is the small w. Let's see what will be the gametes. Here, what kind of gametes can form? One X chromosome is having small y, small w. Another X chromosome that is having small y plus, small w plus. Okay. And if the crossing over is occur, then what will be the result? If crossing over occur, then this is the X chromosome, and that is having one small y, small w plus. And if this is coming to this, then small y plus and small w. Okay, so these are the gametes can form in case of your F2 generation ratio from this individual. So these are the gametes which can form from this individual. Then how many types of gamete can form here? Here two types of gamete can only form. One is the X and another is Y, and this is having small y, small w, and here that is absent. Okay, so let's see for the result, or let's cross between them. See here, if it is fused with this, then what will be the result? Small x, X chromosome, this is small y, small w, this is small y, small w. If it is crossed with this, then what will be the result? X, and this is small y plus, small w plus, small y, small w. Okay, and if it is crossed with this, then what will be the result? This is X, this is Y, and this will be the small y, small w, and this is absent. If it is crossed with this, then what will be the result? X and Y, and here X, there is Y plus W plus. Okay. If we cross between this and this, if we cross between this and this, then what will be the result? X and X, and small y, small w plus, and this is small y w. Then if it is crossed with this or fused with this, then XX, this is small y plus W, and here small y, small w. Then if it is crossed with this, then small y, small w plus x, and this is y. If it is with this, then the result will be the small y plus small w, x and y. Okay, so see here, these are the parental quality or parental varieties. These are the parental varieties. And these are the recombinant one. Okay, these are the recombinant. Here, small y, small w plus, that is not present in the parental generation. Here, small y plus and small w, that is also not present in the parental generation. Here also small w plus and the small y, that is not present in the parental generation. That's why these are the parental generation and these are the recombinant varieties expressed in the F2 generation. And Mendel found that here in this cross there is 98.7% of parental generation or parental varieties are 98.7%. And he found only 1.3% of recombinant varieties. So in this way, he crossed the hybrid cross 2, the hybrid cross 3, that is also performed in this similar manner. Okay, and he found several parental as well as the recombinant percentage in different cross. In the similar way, he performed all these 3 hybrid cross, and the hybrid cross 2, the hybrid cross 2, that is between the eye color and wing size. Eye color and wing size. He found parental combination, that is 62.8%, and recombinant percentage, that is 37.2%. Okay, and the hybrid cross 3, the hybrid cross 3, that is between the body color and wing size, body color and wing size. Here we got parental generation, that is 61.5%, and recombinant generation, that is 37.5%. Sorry, 38.5%. Okay, so here in different hybrid cross, to found different recombinant percentage. Okay, in all these 3 hybrid cross, Morgan found 3 different recombination percentage. That means he come to a conclusion that there may be a chances that the position of the gene may lead to this type of variation. Okay, if the position between the genes are more, then the recombination percentage is the more. Okay, that means the recombination percentage is directly proportional to the distance between the gene. Distance between the genes. If there is more distance between the genes, then there is more chances of crossing over. If more crossing over, then the recombination percentage is more and the linkage percentage will become less. Okay, this is the conclusion of the Morgan's experiment. What is that? He found different recombination percentage in different hybrid cross. Okay, why? He come to a conclusion that the recombination percentage is less or recombination percent vary due to what? Due to the distance between the genes. If the distance between the genes are more, then the recombination percentage is more. If the distance between the genes are less, the recombination percentage is less. Okay, that means that is directly proportional to the distance between the genes. After completion of this experiment, one of the students of Morgan, that is Student, he found another concept. He discovered another concept, that is chromosomal mapping concept. Okay, or genes positioning. Okay, he positioned the genes where is that gene actually present? What is the locus of that gene is present that is performed by stud event? Let's see what happened or what kind of things is given by stud event, that is chromosomal mapping. Okay, let's go to the chromosomal mapping. Let's see chromosomal mapping concept. After completion of the Morgan's experiment, one of the student of Morgan, that is stud event, he gave the concept chromosomal mapping or gene positioning. If we know the distance between the gene, then we can position the gene. Okay, what is the gene that responsible for a particular character, or there is several genes are present over the same chromosome. So if we know the recombination percentage, according to Morgan, if there is more recombination percentage, then the distance between the gene is more. Or in alternate manner, if the distance between the gene is more, then the recombination percentage is more. Okay, and students utilize this concept and come to a new concept, that is chromosomal mapping. Okay, according to Morgan's, the body color and wing size, the genes responsible for the body color and wing size. Here the recombination percentage is more, that is 38.5%. Okay, suppose this is the chromosome. Let's see how this positioning is actually done by stud event. Here the recombination percentage is more. Okay, the 1% of recombination is equal to 1 centimorgan. This is the unit of the distance between the gene. Okay, here 38.5% means the gene responsible for the body color and the gene responsible for the wing size, that is M, this is Y. They have the distance, that is 38. Suppose this is Y. Here this is more, so that I have taken this first. Small y and small m. The distance between these two genes, that is 38.5 centimorgan. Okay? And if we see the, as compared to this is less, and that is 37.2. Okay? Between the gene responsible for eye color and gene responsible for the wing size. The gene responsible for eye color, that is small w. Okay, and this is the wing size, and the distances, that is 37.2 centimorgan. Okay, then if our concept according to Morgan or according to stud event, if the concept is right, then this should be what? We have to minus this 37.2 from this 38.5 centimorgan, and we will get the 1.3 centimorgan. Yes, see here, this is 1.3%. 1.3% means 1.3 centimorgan. Okay, so this is the chromosomal mapping concept. If we know the recombination percentage, then we can positioning the gene. Okay, we can know the gene position. This is about your chromosomal mapping concept. Okay, and this concept was given by the stud event, that is the student of Morgan. Okay, hope you understand this clearly. In the upcoming class, we will discuss other concept of this linkage. Thank you. Hope you enjoyed this lecture. Please like my channel, share and subscribe. Thank you.